Well, I will talk about something that is actually the picture on the left side of the slide. It's very simple, it's very technical stuff, but you will see there is a lot of interaction between the technology and actually the previous presentation that we have seen with the Solvonis and Meteorfarm. So the interesting fact about the technology that was actually patented and developed by the company, by our company, is actually a Mesoporous polymeric material which has a very light surface area and help you in developing something that will address the pharmacokinetics of the drugs without impacting the pharmacodynamic itself. So Brenta is a company that is located in the northeast of Italy and is a fully founded family company. What we're going to do right now is really just to show you something very practical cases. So not really very fancy, but what we will look on is actually the repositioning and repurposing or some typical, very common API that are on the prescription area. One is actually an antibiotic area. We will show you how we were able to increase and have an effective increase of the potency of the drug and remarkable biofilm eradication, some of the issues that we have seen actually previously. The second topic is going to be about diuretics. Diuretics in the way that we were able, together with the example of anti-inflammatory one, to have a very high loading efficiency with our Mesoporous material and with the ability to stabilize in a very stable amorphous form the API inside the Mesoporous structure, and at the same time to increase the aspect of solubility and permeability of the drug substances. So something that is addresses to the pharmacokinetics of the drugs. So the point is that we believe in ourselves. We have a very few handful of competitors, but we were able to translate something from the academia to a definitely GMP, good manufacturing environment and industrial scales. So first example. The first example is related to the application of a specific couple of antibiotics. The first one is a nitrofurantoin, a well-known antibiotic for genitourinary application. And the second one is a non-antibiotic. It's something that has effect as a mucolytic. It is called N-acetylcysteine. What we have seen that when we combine the two drugs together, and we will show you the data right now, we have a dramatic increase of the potency to the versus the drug itself used alone. And we have a platform combination that allows so practically 3 level of magnitude, 1,000 times of increase of the potency of the drug. And at the same time, we have a biofilm rock reduction, which is around the number of 6. So how we observe that? So first of all, the fact that the key figures are related to the, let's say, overall aspect of the antibiotic, and let's say antimicrobial resistance, which is a global crisis, of course. Every year we have more than 40.3 million of people, and we do expect that actually in 2050, the WHO told us that is something that will affect at least 10 million to 20 million people per year. So the challenges are related to the fact that you have most of part of the antimicrobial resistance is related to non-single, let's say, microbial infection, but a complex structure of biofilm that is actually difficult to eradicate. And most part of the drug antibiotics used right now, they are very low permeable to the biofilm inside in structure. And then the third aspect is that most part of the antibiotic, they have a very low solubility, so they are difficult to formulate. They are difficult to deliver to the target of the biofilm, which is the structure that we want to address. So the point is that frequently you use very high dosage of these antibiotics that actually help to increase the antimicrobial resistance mechanism by the biofilm. And sometimes you need also in the same time at the same strength to have a very frequent administration of the drugs. So what we have done, it was simply to understand from a microscopic level how we can handle the fact that we could carry the antibiotic into the Mesoporous structure. So what we have done is really just to build up a polymeric structure around the biofilm, the antibiotics. And as you can see in the structure, in the picture that is actually on the right side, This is actually the example of how the white particle of the Mesoporous platform from Brenta are attached to the biofilm. So this is basically an electrostatic behavior between the structure of the platform to the biofilm. This is the first stage of the mechanism. The close relationship between the biofilm, the structure of the biofilm, and the Mesoporous structure allow the single transfer of the antibiotic into the cell and help to not deliver in the extracellular matrix the antibiotics and let the antibiotics available for, let's say, enzymatic destruction by the biofilm itself. So this is actually something that from a mechanical point of view helps to have the biofilm disintegration, have in the same kinds to have an effective antibiotic releasing side, potentially reducing the dosage and the number of the administration that you have upon the biofilm. Let's see what's happened actually. So we compare the log reduction of the Klebsiella pneumoniae biofilm eradication in synergy with the platform and with a combination of drugs. So the first one, that was the first observation that we have on a standalone non-encapsulated nitrofurantoin effect on the biofilm. When you charge the biofilm into the biofilm, the loaded encapsulated nitrofurantoin in our platform, you have at least one order from two order magnitudes of the potency of the drug that you have seen, that you can see on the slide. So it means that potentially you can reduce not really the strength of the product, but the quantity of the antibiotic that you administer to the overall biofilm structure. When you combine into the platform one antibiotic with a second compounds which help you to disaggregate the biofilm, then you have increased the, you can see you increase the potency of the drug product combination to this synergy by 3 orders of magnitudes. And that's potentially will reduce of 1,000 the amount of drugs in this in vitro section of the eradication of the biofilm, reaching what the biologists call the 6th level of log reduction, so a complete eradication of the biofilm. We tested this kind of application in several different kinds of antibiotics with consistency, with different kind also of the biofilm formation, with the same kind of consistency. So there is first a mechanical principle. In the second, there is the specific delivery on the antibiotics into the biofilm. We don't change the pharmacodynamic of the antibiotics. The antibiotic still works with the same mechanism, but we increase the number of the quantity of the effective antibiotics that is actually delivered into the biofilm. So, talking about the repositioning and the proposing, of course, we have been testing this on the Klebsiella pneumoniae. So if we just look to the community-acquired pneumonia, we can change a typical application of oral administration of the nitrofurantoin to a more effective, let's say, inhaled administration. This is because Brenta works on submicron nanostructure Mesoporous delivery system. So something that will help actually to deliver a product to an inhaled administration. Secondly, topical infection, cause of delivery that are represented. You can move from a typical oral, let's say, application to a direct application of the antibiotic into the biofilm, making use of the sustantivity that the platform help you in attaching the antibiotics into the biofilm structure. Third one, of course, it is very interesting to test different kind of antibiotics in infection of immunocompromised patients. So allowing to have something that is improved in bioavailability and of course has the purpose to deliver the antibiotics via different kind of administration routes, which in the case of immunocompromised patients is something that is very difficult. So second case, second practical case, different kind of a therapeutical area, diuretics. We use a compound very old but very effective, the furosemide, in which we have been seeing that together with the platform we were able to at least load more than 70% of the drug substance into the platform. We were able to increase 4 times the solubility and at least the 50% of the permeability of the product. Why are we talking about permeability and solubility? Because in fact this is one of the key aspects about furosemide. So the point is that furosemide is, for example, used in congestive heart failure. This is something that can affect 50% to 20% of patients per year. In case of pulmonary edema, we have, of course, unfortunately, 40% of people that die in the first year of the treatments. And furosemide is actually used as a primary agent in this case. And with all the side effects related to different kind of pathology affected by the same kind of congestive heart failure. Is a compound which is in the biopharmaceutical classification system arena is probably one of the worst to formulate. It's very low solubility, a very low permeability, even for the fact that it is a very high effective drugs. And traditionally, if you want just to bring the furosemide as a drug substance itself, you go through a very complex pharmaceutical process prior to make the drug products, which is usually go to increase the surface area of the drug substance, of the API, through micronization process. A micronization process actually is a very long process. It's highly complex in my point of view. Not all the companies are able to make it, since that requires specific handling. And it's a top-down technology, whereas the Brenta platform is actually a bottom-up technology that helps you to size the exact dimension of the surface area in order to reach a specific particle size distribution. So what we have done is try just to see if we were able to impact the formulation, get a better performance, a scalability of the process itself, giving to our partner the opportunity to have a drug product intermediate that can be directly formulated. So we compare what's happened if we encapsulate in the same way that we have seen the picture before with the nitrofurantoin in 2 different polymeric Brenta platform, the furosemide, and compare the solubility with the standard requirement from the European and the US pharmacopoeia, so pH 5.8 and pH 6.8. As you can see, dark blue, the furosemide solubility over the time, and the improvement that we have in the green line and in the light blue of the furosemide dissolution profile when it's actually encapsulated into the Mesoporous platform. So we can have up to 4 times, as I said before, of an increase of solubility, especially in a short-term, let's say, availability of the furosemide. What you would expect, of course, if you have an increase of solubility, could you expect that the more quantity of drug is making available to the organism? So this is something that we have demonstrated in vitro. We tested later on the increased solubility to get an increase of permeability related to the Fick effect. So the fact that if you increase the solubility of a given drug substance in the local point of administration, it could have an increase of the osmotic pressure of the drug substance to be absorbed in a specific area of absorption of the drug substance. And this is what we have seen in a simple, very simple permeability testing, that the furosemide, when it's encapsulated in the first stage after 1 hour, can have an increase after 4 hours at least of the 50% of the overall permeability compared to the free furosemide, which is actually in this test was actually the furosemide already micronized from the supplier that we selected from the drug substance. So the point is that we observe an increase, a linear increase of improvement of permeability of the drug substance in this way. So a point of view of the repurposing, what are the key let's say, strategic aspect that we expect on the use of furosemide. So that we have, for sure, an increased homogeneity of the drug substance into the formulation. And this has actually helped, as you can see, to have a consistency in the delivery of the drug substance from the formulation. In the same time, you can keep your oral administration as a point of view of, let's say, the drug application, and change the drug oral formulation with something that has a very rapid onset, like oral film or oral disintegrating tablets, something that is actually not available right now into the market, because the specific property of the furosemide. Third case, a completely different kind of compound. So first antibiotics, second furosemide as a diuretic. Here we will look to anti-inflammatory compounds. So a complete different therapeutical point of view. This is just to show you how versatile could be really the platform itself to be used. We reach with the indomethacin, which is an anti-inflammatory compound, the 85% of loading efficiency, at least an 11 times of increase of solubility, and a very dramatic increase also from the point of view of the permeability in the same way that we observed on the furosemide. So indomethacin is a very common anti-inflammatory product, very old one. I think it has been approved during the 60s of the last century. So it has a very long history in point of view of safety and efficacy itself. But as a compound which is belonging the, let's say, NSAID and the biopharmaceutical classification system class 2, together with the fact that we can modulate the solubility, has for sure a very low permeability. And this is actually one of the common challenges of many of the anti-inflammatory drugs. So the fact that having very low permeability, in the same time, they could be addressed to a lot of side effects, which include the so-called black box warning in the label of the products, cause the cardiovascular and kidney complication, gastrointestinal irritations. So the fact that if we improve the solubility and the permeability itself, we can have an optimized pharmacokinetics, and we can have potentially the opportunity to remove the black box warning and have a more safe use of an anti-inflammatory compound. So what we tested in the same way, we encapsulate the indomethacin in two different polymerase structures patented by Brenta. One is actually, as you can see, tested at two different pH, and from the first point we get a dramatically increase as before of the solubility of the product. And at the same time, we tested the permeability aspect, which were, at least as I show you, compared to the free indomethacin, at least 44% higher than the free indomethacin one. So point of view of the repositioning, you can have an expansion of the therapeutic option. Is that possible that you can move something that usually is done in the prescription way, similarly to other anti-inflammatory drugs in the OTC environment, because you decrease the amounts of the effective drugs in your formulation? By lowering the dose, you can remove the black box warning, which is always something interesting to look and have much more available to the prescription, the products, and also to have a market expansion that we would expect could affect the different kind of anti-inflammatory drugs. So the major takeaway about the technology platform in cell, with this one I'm going to finish, we work on the pharmacokinetics. We have a proprietary platform. We don't affect the pharmacodynamic for your drugs. Your drugs will keep the effector and the biological behavior as it was before. We work on the pharmacokinetics. We work increasing solubility and permeability in the first instance. We work on the opportunity to make more effective drugs, reducing the number of dosage and possibly the number of intakes that you have per day. So something that actually, knowing better solubility and permeability, even help the first stage of development and the development of formulations. We increase the homogeneity to the end on the drug products. Potentially, especially when you have a very low dosage compound, very effective compound, increase the compliance with your patents. And to the end, the major aim is always, as any one of us, really just to, let's say, improve, which is the lack of proportion between the side effect and the efficacy of the drugs. And of course, we, as a platform, we tested already, which are the factors related to the reachability and the biocompatibility of the compounds that we make, which virtually belong to the generally recognized as safe material, and that we make available to different kind of application. So this is the end. I'm happy to take your question if you have any, or just came to our table and in a more private way, maybe we can share some of the other topics that you are interested with.